Okay, guys, so we're back now to talk about plants. And as you can see from this slide, we divide the plant kingdom based on how they reproduce, whether it's with seeds or not, and also if there is vascular tissues or not. This lab is going to focus on the bullet point 1 and 2, which are our non-vascular, so no tissues to carry nutrients and water, seedless plants, and our vascular seedless plants. So under our non-vascular seedless plants, we're going to focus on our hepticophyta, which are liverworts, and bryophyta, which are bryophyte mosses, but anthocerophyta hornworts are also in this group. And we're going to focus on vascular seedless plants, pterophyta, which are ferns, and lycophyta, which are club mosses. And then in our next lab, we'll look at the vascular plants that have seeds or cones. So there are some common plant characteristics. They are photoautotrophic, which means that they do photosynthesis in order to get their food. They have cell walls made of cellulose or starch. They are non-modal, so they do not move. Some species do have flagellated sperm, but because gametes are not independent living organisms, plants as a whole do not move. They can reproduce sexually and asexually. They can be monoecious, meaning that they have male and female structures on the same plant. Try to remember that using the prefix mono, meaning one. So one plant that has both male and female structures. Or dioecious, having separate plants. One is male, one is female. Most plants are going to have haploid and diploid multicellular forms, and we call that alternation of generations. In animals, we know only our gametes are haploid, and they don't grow into a structure. So it's very different for plants. In general, we said that plants have alternation of generation, or that they have haploid gametophytes. Phyte meaning plant, gameto gametes. So this is the gamete producing life stage of the plant. It's what's going to have the sex organs. The sex organs are called the antheridium. That's the male sex organ that makes sperm. Or the archegonium. That's the female sex organ that produces eggs. Gametes are going to fuse, and that makes a diploid 2N zygote. And when we have that zygote, it is going to grow then into our diploid. Sometimes you'll see 2N sporophyte, and that is the plant that makes spores. And then through meiosis, that zygote will grow into that sporophyte. That is going to make haploid spores that develop into the gametophyte. So let's start with our bryophytes. They are the most primitive type of plants. They do not have a vascular system, so they have nothing to transport water and nutrients. They have to rely on osmosis and diffusion to move these things throughout the plant. Because of this, it limits how big they can get, and they have to live in environments that are damp. In order to stay sort of connected to the ground or the structures around them, they have rhizoids, which are these root-like sort of hairy structures. This is the most common group of plants after angiosperms. There's about 24,000 known species of bryophytes. And we are going to focus on the moss and liverworts, but hornworts are also in this group. How do bryophytes reproduce? They have a dominant life cycle or a dominant phase in their life cycle of a gametophyte. So when you look at moss, you are looking at a haploid organism. The thallus is what we call the body of a moss. It's like the little um, leaves of the grass-like structure that makes up moss. They have flagellated sperm that has to swim to the eggs. So we know that they have to live in a damp, moist environment. Their diploid zygote is going to grow into this upright stalk sporophyte, and that is going to include something called a seta. That's like the stalky part, a capsule at the end of it, and then an operculum, which is like the little door that keeps the capsule closed until it releases the spores. Meiosis is going to make these haploid spores inside of that diploid sporophyte capsule. And then upon release, these spores are going to mature into the new gametophyte plant. Mosses can also reproduce asexually, doing fragmentation. So you could take moss like off the ground, blend it up in a blender and make, a little ball of moss, toss it somewhere, and it will grow new moss. This slide here shows us our phylum Hepticophyta, which we're going to look at the species called Marchantia. You can see on the bottom picture that flat leaf-like structure is called the thallus, and on it you'll see these little cups called gemi-cups. They are going to hold spores, and so if water gets into these cups and they overflow, the spores are released, and then we have asexual reproduction. The top picture shows us what that zygote will grow into. This is our diploid sporophyte, and we can see the capsules. It sort of looks like a weird little palm tree. That's holding on to these spores that will eventually be released and grow into the thallus. You guys are going to have specimen available for you, or videos of specimen available for you on the D2L. But you can also use these pictures here to look at a Marchantia gemi cup. This is like if we took a cross section or a cut of that cup and laid it flat, and you can see that it's holding on to those spores. And then we also have our Marchantia capsule that is holding onto the spores. This is that thing that looked sort of like a palm tree. This is what that would look like if we cut into it and laid it flat. You'll also see some information about Minim. That's our moss. And you can see here the sort of leafy stuff. That's your thallus. And then growing out of it, that is your capsule holding onto those spores. So you'll be able to see on the D2L a video about the protonema. That is going to be as soon as that spore drops out of the capsule, this is what grows. So it's the beginning stage of the gametophyte generation, and it looks an awful lot like algae. So this is how we know that this moss, these bryophytes are most ancient. They're most closely related to algae. It's how we know algae is the precursor to land plants. You'll also see our Minim sporophyte capsule. This is where we're holding on to those haploid spores that will be released to grow our new moss thallus. But it is that diploid capsule. And you'll see the operculum, that's like the little trap door that is holding the spores in until they're released. Okay, so now we'll move on to our vascular seedless plants, where we have our pterophyta and lycophyta. With our seedless vascular plants, what we mean by vascular is that they have tissue to transport water and nutrients. And those tissues are called xylem and phloem. The way that I use to remember what does what. Phloem, that PH has an F sound. That is the same as the F sound in food. So phloem is moving the nutrients, where xylem, I like to remember WXYZ. It's transporting water. There's your W, XY, and xylem makes the Z sound. If that helps you, great. Feel free to use it. If not, that's okay too. When we're looking at a fern, we are looking at the diploid sporophyte generation, so different than our mosses. We're past being gametophyte dominant. We're now diploid sporophyte dominant. And the most common phylum that we see is Pterophyta, which are our true ferns, but club mosses is our phylum Lycophyta, also lumped in here. And Lycophyta is going to have true roots, true stems, and true leaves. And this is the first time that we will see that in our plant kingdom. So what do we have as our fern structure? The first is we'll have these sporophylls. Those are those leaf-like structures on a fern, like one little leaf of it, where as a whole, the whole blade of the fern is called a frond. You'll see that there are euphylls, also called megaphylls. Those are that there are lots of veins in our true ferns, or one vein in our club mosses. These plants will also have stomata, which are pores that will open and close on the underside of our leaf or frond that is going to allow for gas to exchange, and those are necessary for photosynthesis. You'll also see sori or sorus. Those are going to be these cluster of spores and spore holding tissue on the underside of the leaf. And then we also want to talk about a prothallus. That is the haploid gametophyte. And it's this about 1 millimeter large heart-shaped structure that has rhizoids. And it's where the antheridia and archegonium are. They are on the underside of the prothallus. How do our ferns reproduce? Well, like we said, their dominant form is diploid. So when you look at a fern, you're looking at a two-end structure. And it is going to make diploid spores, which is different than our moss. Those spores are going to undergo meiosis, and then they will do mitosis to make the prothallus. And that again is our gametophyte generation. There is flagellated sperm that has to swim to meet the eggs in the archegonia. When they meet, fertilization takes place and we get our diploid zygote. And that zygote is our new sporophyte that is going to grow into our fern right off of the surface of the prothallus. And here are just some pictures sort of showing you that fern frond. If we flip it over, we can see that sporangia, the sori on the underside of the leaf. That sori is going to be holding on to the spores, and they will release the spores, which will grow into more ferns. So what you're going to find on the D2L are videos showing you the prothallus, that little heart-shaped structure, and showing you the archegonia. That's the female reproductive structure that's sort of found in the middle of that prothallus. And then you'll also see the antheridium that's spread throughout the prothallus. You'll also see the little root-like structure, those rhizoids that hold it in place. And then you'll see some videos from our horsetails or equisetum. They're also sometimes called scouring rush or pop grass. These guys have silica in their structure in their cells, and they were used by pioneers to scrub stuff. So that's why they get the name scouring rush. You'll also see some information about whisk ferns. That's our silatum. These guys are interesting in that they don't have any sort of leaves or leaf-like structures. They are just these stalks that are photosynthetic, and they are native here in South Carolina. You can find them. And then talking about our club mosses, we don't really see any other preserved specimen, though there will be a video for you to look at on the D2L. Their name is sort of confusing, and what they are is a little confusing, because they do have a strobili, this sort of cone-like structure, like our gymnosperms, our cone-bearing plants, but they don't have seeds; they have spores. And as we said before, they're our first plant where we see true leaves, true stems, true roots. Sometimes these guys also get called like ground pines because of this cone and all of those structures. But they don't have seeds; they have spores. That's why they're different. So if it wanted to be a fern or like a fern, it should have been named a club fern, not a club moss, because it has vascular tissue, but it's not a pine. So this is why taxonomy and scientific names are so very important. So here's one of those structures that we were talking about, our lycopodium. You can see the strobili on it, and you can see how it sort of does look like a little ground pine, but it is not. It's in the phylum Lycophyta. So though we are not actually in a classroom, we're doing this online, here's some checklisty type things that you'll need to know about to fill out your lab worksheet. You're going to want to know Marchantia, Minum, Pterophyta, Bryophyta, um, and our different preserved specimen. Again, these are all going to be via videos. But that's pretty much it. Oh, there's some unknowns. So I'll show you guys these, and then we'll sort of pause this video, and I'll leave an answer key for you guys to see how you did. So what is this guy? What should we know about him? And how about her? What's this? Okay, so you can check the D2L for the answer key, or even go back through these slides. These pictures are in there. But if you have any questions, please feel free to let me know. You can use the discussion post, the Remind app, email, office hours. I'm happy to help. Hope you guys have a great day.